Morning to all. We are entering our 13th day of our BIIS-12 workshop. Warm welcome to all of you once again. Today is for our first lecture. We have with us Dr. Nivas Kumar. He is a scientist and a GYTI program coordinator of Srishti. He has his PhD from Department of Environmental Science, IIT IASM, Dhanbad. His research interests are mainly focused on microalgal biofuel production technology. He has published 9 research papers in international reputed journals. He has received a CSIR fellowship from 2014 and also received ICMR fellowship at 2015. He has also received Best Paper Presentation Award from the Association of Microbiologists of India 2018. So with this short introduction, I invite Dr. Nivas Kumar to deliver this lecture. Dr. Nivas, the floor is yours.
Thank you, Dr. Amar. Thank you very much. Good morning to all. So my today's topic is microalgae, a resource for wastewater treatment and biofuel production. So I would be discussing my talk under the following contents. So these all are the contents of my talk. So first we will discuss why microalgae is important. So as we had written in the headlines that microalgae is used for the biofuel production, wastewater treatment. So why microalgae, not any other subject?
Your voice is breaking. Put your mic.
Am I audible?
Yes, now better.
So as we have seen that we have limited land resources. Only about 12% of total ice-free land. It is only used for the agricultural purpose and various activities for humans. So we have a very limited land resources and limited fresh water. Out of the, because we have only less than 3% of fresh water which we can use, and within this 3% fresh water, most of the fresh water is stored in glacial forms or in unused form like in moisture or various other forms. It is only less than 1% of fresh water which is available for the direct use which we can use. So we have a constraints of limited land resources, constraints of limited fresh water, and we are all aware that the pollution is also increasing day to day due to the incessant increase in the human population and utilization of various fossil fuels for human aspirations. And it is also estimated in the prognosed that by the 2050, we will reach the global human population will reach up to 9.9 billion. So due to such a hike in human population, there would be a very huge demand of food, fuel, and fresh water. As you can see, as per the prognosis, 60 to 100% will be a demand of food by 2050, and 50% demand would increase in biofuels, whether it is in biofuel or different. So, due to these constraints, scientists have thought that we should think some alternatives. So, and also we are also aware of various different kind of environmental protections and sustainability due to we are facing in fossil fuels. So, what is happening in today's world? Fossil-based economy is shifting towards the bio-based economy. So, what is fossil fuel economy? So, fossil fuel-based economy is an economy in which various foods and fuels and chemicals we obtain from fossil fuels either directly or indirectly. And we all know that these fossil fuels are very hazardous for humans, health, environment, and various other aspects of the earth. So, what is happening in bio-based economy? So, we use biomasses. Biomasses biomasses for the aspirations of different type of fuels and chemicals. So, when we are getting any things like food, fuels, and chemicals from biomass, so there is a increase in food prices because biomass is also a staple source of food for human and related animals. So we have to think about that if we continuously use biomass for the boosting of economy, then there would be a very much hike and fight for the food prices of the food and various other bio biomass. So we have to think about the alternatives of biomass, like agricultural biomass. So in this constant, microalgae stood very good for the production of these things. Why microalgae is very good for this type of thing? Because microalgae do not require any arable land for cultivation. Because if you have to cultivate any food grains, and as we have said in this slide, that we have only limited land resources. So if we use agricultural land for the production of biomass, which is intended for the production of chemicals, fuels, and food, then there would be a competition for food and other bio-based products. So microalgae has a benefit that it do not require any arable land for the production of biomass. Second is that it can be cultivated in seawater and wastewater and do not require much fresh water. Second is that the biomass productivity of microalgae is very much higher than the agricultural crops. They are also a valuable source of different type of uh, pigments like lipid, like chlorophyll, like astaxanthin, like carotene, like carotenoids, and different pharmaceutical and nutraceutical products. And one important thing is that microalgae has a very higher sequestration property, carbon dioxide sequestration property, which is almost 10 times much higher than the terrestrial plant. So microalgae is very important. So we are using microalgae for the production as well as the treatment of waste. So let's look what is microalgae. So microalgae is a microscopic organism that belongs to both domain of life, like prokaryotic or eukaryotic. It is basically a aquatic body, aquatic microorganisms, and it can remain in marine, freshwater, and soil also. And it has a surface charge potential, which is almost negative charge. Surface charge we will discuss in the coming slides. What is surface charge? And they are mostly photosynthetic, and they are responsible for around 50% of global oxygen production. So let's talk about what is beta potential or surface charge potential. So microalgae contain different type of electrokinetic groups in their cell wall, like amines, like hydroxyl group, like ketone group, and so many groups are present. And these groups at different pH specify a negative charge. That is why the surface charge of microalgae is always negative. So if you look here, so when microalgae comes in a liquid or any conducting liquid, like you can ionic liquid, you can think, or water. Water is also conductive liquid. Then what happens? The negatively charged kinetic charges attracts counter ions, like microalgae is negatively charged, then it will attract positively charged bodies to their surface, and this stick to the microalgae. And this layer, which you can see here, this is called the stern layer, which is directly attached to the microalgae particles. And this, apart from this layer, there is a diffuse layer which contains different type of ions, like positive and negative ones. And these two layers, which we call diffuse layer and stern layer, is called double layer. So when a microalgae will propagate in liquid, it always contains two layers. One is stern layer and another is diffuse layer. So and this diffuse layer is when the microalgae move in the bulk medium, this two layer move with the microalgae. So there is a slipping plane exists at the diffuse layer and bulk medium, and the potential at this diffuse layer or the slipping plane is known as the theta potential, which is very much important in microalgae biotechnology. Now, different type of microalgae. So we have a document said that around 200,000 to 800,000 algae species exist on the earth, and out of which 40,000 species has been identified. So microalgae belongs to different type of groups. You can see chlorophyceae, which is highly used for different activities like wastewater and biofuel production. And some of the examples you can see here. Then Purification, it is also known as the brown algae. They are mostly marine, and unlike Purification, chlorophyceae are marine and terrestrial also. But all these rhodophyceae, cyanophyceae, these all are basically marine microalgae. So another is bacillophyceae and chrysophyceae. These all are the microalgae which are marine, but they are planktonic photosynthetic microalgae, which controls the food chain and food web of the oceanic system and our marine system. So these are the different kinds of microalgae exist on the earth. So if you want to use microalgae for any kind of activities like wastewater treatment, biofuel production, or pigment production, or any other production, you have to first isolate microalgae, because in the nature, microalgae is not pure form. It will contain some consortium of microalgae or fungi. So to get its proper benefit, you have to first isolate the microalgae from the nature. So there are different steps that we follow for the isolation of microalgae, which starts from the sampling. First, we go to the desired space centers where we want to obtain microalgae. Like we want to obtain a microalgae for wastewater. So we will sample it from the wastewater, usually because microalgae existing or living in the wastewater are totally adapted to wastewater. So we will sample it from wastewater. If we want to go for marine wastewater treatment, marine treatment, then we will sample it from the marine environment. So after sampling, we will go for the removal of contaminants, because microalgae contain different types of contaminants like bacteria, fungi, and the mycofungi. There are so many type of microorganisms who cohabitate with the microalgae. So we will first remove the contaminants through filtration. Then we will go for the sample enrichment, because we have sampled the microalgae from the natural environment. Its concentration is very low, so we have to go for enrichment. So after enrichment, we will go for the sample purification. So in fact, sample purification will be used for antimicrobial agents, including antibiotics and antifungus. So after these kind of pre-treatment, we will go for pure isolation of microalgae using a definite media. So one of the media is tap media, which is also known as tris acetate phosphate media, which is used for the culturing of any kind of microalgae, because it contains all the nutrients available required for the microalgae. And then after the microalgae, we have to go for it to study its different type of photoperiodic behaviors. And so after the isolation and then culturing in a proper nutrient media, we will go for incubation. So these are the steps that we follow. And here you can see that what we have isolated microalgae in consortium. Then after serial dilution, then strict gating, we will get a pure colony. And this pure colony will be then resampled and then cultured in higher volumes. And then finally we culture and maintain in algal culturing units. Then let's talk about wastewater. So we have just seen what are the microalgae and how to isolate. Then if we want to treat wastewater, then we have to know what is wastewater. So wastewater is any kind of polluted wastewater generated from the human act. So there are various components which exist in the wastewater, like organic components or inorganic components. You can see that carbohydrate, proteins, grease, oil, pesticides, agroeffluents, and antibiotics. These are all of the organic components of the wastewater that usually present in any kind of wastewater. Then in inorganic contents, which will contain heavy metals, calcium, magnesium, chlorides, and other ions. So different type of wastewater. So we have different type of wastewater, but we can divide into basically three type of wastewater, which is industrial wastewater, municipal wastewater, and agricultural wastewater. So if you are getting from any type of inputs from any industries like pharmaceutical, petroleum, tannery, paper, then it will industrial wastewater. And if you are getting effluents from the household or domestic users, then it is a municipal wastewater. And agricultural wastewater, then if you are getting a runoff from agricultural farms, slaughterhouses, dairy, piggery, it will comes under the agricultural wastewater. So what distinguishes with agricultural wastewater or municipal wastewater among them? So if you have the baseline of distinguishing whether it is industrial wastewater or municipal wastewater is the organic strength. If you have an organic strength more than 100 mg per ml, then it is industrial wastewater. And if organic strength is between 200 to 800 mg per liter, then it is basically comes under municipal wastewater. And if it is a moderate between, it could be less than 200 or 500, then it is not a very hard and fast rule, but it is, if this criteria is fulfilled, then we can say that this comes to under municipal wastewater or agricultural wastewater. So these all what are the nutrients? So these all are the nutrients which are available in the which could be present in the different type of wastewater. So one question is that why we treat wastewater? So you can see that. So if we can reuse this much amount of wastewater, then we can recover 2 into 10 to the power 10 5 tons per year of nitrogen and phosphorus. So we can recover various kind of nutrients like nitrogen and phosphorus. As you can see in the table, like in different wastewater effluents, various type of nutrients are present in different types. And this is the table showing you the comparative tables for different elements, which are the micro elements which are present in the wastewater. And these micro elements are very beneficial for the cultivation of microalgae. That's why microalgae is very used to when we cultivate in the microalgae, because it contains all the nutrients and micro elements present in from the wastewater. So, and these all are these are the different type of cultivation system that are used for cultivation of microalgae. So it is a raceway pond, you can see, then circular pond, then unswept pond. So what is different between the raceway pond and circular pond is that raceway pond has rectangular surfaces and circular pond has a circular diameter, you can see. So this, and the upper phase content shows the open system and the lower one shows the closed system. So the main difference is closed system and open system is that you have a very negligible chance of contamination of in closed system, because the system is very closed. If you are injecting only a single species of microalgae, then it will be a single species in closed system. But in open system, it is a very high chance of contamination, because if you are injecting a single micro species of algae, then after the passage of time, you can you will sample and we visualize it under the microscope, you will see that different microalgae have colonized due to open system, because open system takes different type of inputs from the air, from the soil, and from the winds. So the open system is not so good for the cultivation if we are intending for extraction of any kind of products from single species of microbe. So for specific product abstraction, we usually go for closed system. Then what are the mechanisms that microalgae use for the treatment of wastewater? So microalgae use different type of mechanisms, which comes under the physical, then chemical, and then physiological mechanism. So precipitation. So microalgae, it is a species-specific mechanism. So every microalgae will precipitate different kind of heavy metals, microbes. So it is a non-specific technique of microalgae wastewater treatment. And it is basically based on the charge and different type of polymer that microalgae secretes. So what happens? Microalgae secrete charged particles. Like in polymers, they are always charged. So this charged particle attract different kind of particles and then entrap it into a precipitation. Then simple adsorption. Adsorption, you all know, it is a surface phenomena. So different kind of micro, like viruses, bacteria, usually get stick to the surface of the microalgae. Then another is the biotransformation. So what happens in biotransformation? Microalgae just intake metals or different kind of nutrients, and it goes into the vesicles or different vacuoles of the microalgae, where they transform through the enzymatic actions or metabolic action and transform into different kind of products or particles. So this is the another kind of mechanism. Then the bioaccumulation. In bioaccumulation, what happens? Microalgae will not totally metabolize these substances. They first accumulate these substances in higher concentration, and its fate will be, it can be biodegraded or biotransformation. Then mechanism of nutrient removal. So nutrient removal is different, and this is a non-type of nutrient removal. So if we're talking about the phosphate removal from the wastewater, then if the phosphate is an organic phosphate, then it will comes inside the microalgae through a different kind of channels, which is known as phosphate channel, phosphatase channel. And this phosphatase channel changes organic phosphorus into phosphate, hydrogen phosphate. And this hydrogen phosphate through through metabolism of microalgae can get converted into ADP and ATP, which we use for energy currency. And if microalgae, if it is in phosphate form, then it will not go to phosphatase. It will use it comes under the microalgae body inside using transport proteins, and these proteins just enter this phosphate into microalgae cells, microalgae internal environment, and convert into ATP and ATP and nucleic acids. And if they have excess of phosphate, then this phosphate is generally stored into polyphosphate granules. And these polyphosphate granules are very important for microalgae bio abstraction of phosphate from the wastewater. Then nitrate, ammonia, this all comes under the microalgae through different routes. And if it is nitrate, then it will come through transport protein, and it will change into nitrite, then ammonia. And if it is only ammonia, then it will directly go inside the cell surface, cell internal environment, and gets converted into proteins. And then carbon dioxide, it is converted into sugar and starch. So microalgae just take these substances and convert into different usable forms. So if you are using microalgae, so first after treatment of my microalgae, you have to harvest the microalgae. If you want to take any kind of resource from microalgae, then first we have to harvest the microalgae. Harvesting is generally microalgae basically remains in suspension, and as I said earlier in the earlier slides, microalgae are usually charged particles. So if a suspension isn't are closely approaching, then they will repulse each other. So the surface, so the culture of microalgae is always static. It will not allow the microalgae to congregate itself because they have a surface charge, negative surface charge, and these negative surface charge repulse each other. So the microalgae is always a static charge in the suspension form. So harvesting is basically the removal of microalgae in concentrated form from its culture condition, whether it is in wastewater system or whether it is in artificial culture. So it is basically the congregation and then removal from the water system. So harvesting usually costs around 20 to 30% of total biomass production, because of charged nature of the microalgae. We have to put extra effort to harvest the microalgae. So harvesting is so costly because it's less size, then it's less concentration, and then presence of heavy theta charge potential. So we use different kind of harvesting techniques, which comes under biological, physical, and chemical harvesting techniques. So as you can see, in biological techniques, we use autoflocculation, then fungal or bacterial assisted harvesting techniques. In physical, we go for sedimentation, ultraflocculation, and acoustic systems. But among them, the chemical method is very prominent because it is non-specific. And in chemical methods, we use generally coagulation or flocculation. So flocculation, we have to use a flocculant or coagulant that is used for the harvesting. So as we have seen, that microalgae are charged particles. So if we use a counterion, a polymer or a substance or any chemical which contains charge opposite of the microalgae, then it will reduce the charge, apparent surface charge of the microalgae, and allow the microalgae to congregate. So for this purpose, we use different types of organic chemicals. So mainly organic polymers, and then we use synthetic, which comes under synthetic or natural polymers. Some of them are polydadmac, PEI, chitosan, starch, which are generally used for the harvesting of microalgae. Then in organic microalgae, we generally go for the metallic salts flocculants like alum, polyammonium chloride, magnesium chloride. These are all of the salts. But one thing is that inorganic chemicals are not good for the microalgae harvesting. Even we are used when we are targeting the production of microalgae for human consumption, because these particles, these ions are sometimes become toxic to humans and human-related animals. So we generally go for the natural polymers that contains different type of different kind of charge. So for polymeric flocculants, we can go for natural polymer-based flocculants. So this slide shows the preparation of natural polymeric flocculant. So in this slide, crude polymer of locust bean gum has been cationized using etherification reaction. So in etherification, what happens when this LPG polymers are very tough in nature. So if you will dissolve in water, it will not dissolve. So for dissolution, you have to use NaOH, and we use heat. And under the action of NaOH and heat, this polymer become very loosened. And due to this loosening of the polymer, these reagents, GTMAG, which is the full form of GTMAG, is gradual trimethyl ammonium chloride, which contains a charged ammonium group. And we use it in a very less concentration, that it will not harm, or it is not harmful for any human or any living organism. So under these settings, these GTMAG get attached to these polymers and provide them a positive charge, less positive charge, so that this can neutralize the surface charge of microalgae, and ultimately the microalgae would be get harvested. And after the polymer synthesis, we just go for the characterization. So there are some characterization which we can do. This is the characterization result of FTIR. As you can see that in cationic polymer, there is a specific band is present. This is 1477, and which is only present in GTMAG, and which is absent in the locust bean polymer. So the appearance of this band after the cationization shows that our polymer has been got cationized. So this specific band is we use for the identification of ammonium group insertion in the polymer. And as you can see in the FESM, which is field emissions scanning rate electron microgram graphs, you can see that this LPG before the cationization was smooth, oval and round in shape, but after the cationization, it becomes irregular and very distorted. So these two characterization shows that the surface of the polymer has been got cationized or distorted. So this signifies the cationization or insertion of positive charge. And then we also go for zeta potential measurement of this polymer. So if we will measure the zeta potential before and after the cationization, we will get a positive zeta potential, and this positive zeta potential would come from after the cationization of the polymers with GTMAG. Then you can see that this polymer, how we use for the harvesting of microalgae. So we take microalgae, then add a definite amount of polymer, optimized amount of polymer, and then we will go for mixing. And what mixing does, mixing will neutralize the apparent charges of the microalgae, and ultimately it will cause flocks. After flocculation, this microalgae, and you can see in this picture, in this microscopic image, before the harvesting, the microalgae are very spacey, there is a very much distance between them. And after the flocculation, they are aggregated. There is no space between them. So these are the benefits. So you can see in this chart that in very less concentration, at a dose of 55 ppm, these microalgae got harvested. So ppm is a very less concentration. And so after the harvesting, we go for the extraction of the various type of products from the microalgae, like biofuels. And biofuels constitute different type of fuels like biodiesel, bio ethanol, and biohydrogen. And these all are the renewable sources. Why it is renewable sources? Because microalgae is a renewable body. We can renew it within 15 days or 14 days, and it will lead to a zero emission of carbon dioxide, because carbon dioxide which will be generated after the ignition of biodiesel, which has already been used up during the cultivation of microalgae. So the microalgae from the biodiesel or the biofuel the CO2 that will be exhausted, it has already been abstracting from the environment. So it is known as the zero emission carbon dioxide biofuel. So biodiesel is generally, there are some steps that we follow for the production of biodiesel. Just we go for the microalgal culturing. Then after the biomass harvesting, we will go for the lipid extraction. For lipid extraction, we can use chemical and mechanical methods. And after the chemical lipid extraction, we would go for the trans-esterification because microalgae, uh, because lipids are very heavy in nature and it is very polar. So what trans-esterification does, trans-esterification uh, create a very moderate polar substance. And you can see here in this figure, the trans-esterification of the lipids, where the triglycerol has been changed into the biodiesel of fatty acid methyl esters. And after the trans-esterification, you will get glycerin bioproduct and then fatty acid methyl esters. So production of biodiesel through extraction of lipids from microalgal biomass and conversion into lipids into biodiesel through catalytic and independent process. So trans-esterification can be catalytic dependent or catalytic independent processes. So for the catalytic dependent, we use chemical and biological enzymes for the catalysis. So we would definitely use lipases. Lipases are the enzymes which we use for the biological catalysis of triglycerides into biodiesel. So then after the biodiesel, we can also obtain bioethanol and biohydrogen. So microalgae generally accumulate very huge concentration of carbohydrates. These carbohydrates, depending upon the microalgal species, could vary from 21% to 64%. And these carbohydrates are generally used for the production of biofuel. So what we use, the harvested biomass is first go for the pre-treatment. We use autoclaving, enzymatic pretreatment, sonification, enzymatic pretreatment, and then acidic pretreatment for the biomass pretreatment. So what does biotreatment does? Biotreatment will just convert your complex sugar into less complex sugar. And this less complex sugar will be the input point for the production of bioethanol. So after the conversion of complex sugar to less complex starch or sugar, we use different kind of bacteria or yeast. Generally, we use Saccharomyces cerevisiae for the production of for the fermentation of this less complex starch into ethanol. And after the purification of this ethanol, we can get a purified form of bioethanol. And then we can also obtain biohydrogen. So biohydrogen is a carbon neutral fuel because after the ignition of hydrogen, it will only produce water. No carbon dioxide is produced. And so it is a very purest form of fuel which we use, which we can get from the microalgae. So after the biomass harvesting, we can obtain biohydrogen through two processes, either through biophotolysis of water or starch fermentation. So what happens during biophotolysis? So microalgae have a photosystem, which contains two types of photosystems, like photosystem 1 and photosystem 2. And photosystem 2 has a potential for the conversion of water into oxygen and hydrogen. So this biophotolysis, we get hydrogen. And then another process through which we can get biohydrogen from microalgae is the starch fermentation. Because as we have seen earlier in this slide, that microalgae contains different very high concentration of some microalgae, if I remember, chlorella, and another is the Senadesmus. These two species have a very high content of carbohydrate, and this carbohydrate or starch, we usually go for the fermentation, and this fermentation we get biohydrogen. So and this is a summary of how we can use microalgae is used for the wastewater treatment and production of different types of biofuels. So first we go for treatment, then we would go for the microalgae cultivation system. We will cultivate microalgae in different cultivation system like open, close, and different cultivation system. And then in this cultivation system, microalgae will interact with different kind of wastewater components and use different type of mechanisms for the extraction and abstraction of nutrients or different type of pollutants from the wastewater, and ultimately the microalgae will clean this wastewater and treat this wastewater, and this treated wastewater could be used for the industrial or agricultural use, and the biomass that we obtain from the microalgae, which we can use for the production of biodiesel, biohydrogen, bioethanol, and as I said earlier, the microalgae are very good source of pigments, natural lipids, nutritional lipids, and biogenic polymers. So all these things are we can just obtain after the microalgae harvesting. So microalgae does two type of different kind of biotechnological potential, and one of them is purification of wastewater or treatment of wastewater, and another is the production of valuable biomass that we have seen. That's all.
Thank you, Dr. Nivas Kumar. The session is open for discussion. You can ask any questions or doubts you have, you can ask Dr. Nivas. Questions? Dr. Nivas, there is a question in the chat box.
या वंचन भाई ये हमारे
Or somebody has asked about the blending of bioethanol.
Yes.
That is the question.
What is the scope of blending of bioethanol with the normal, I think, with the petrol or diesel? Yes.
Yes, sir. I want to ask, like, how efficient is this and with microalgae?
The government of India is starting an initiative for blending of up to 10 to 15% of bioethanol in different kind of diesels. So we have to, in this way we can reduce the dependency of different oil producing countries. So open a very huge scope of jobs and different kind of and also open different non-polluting sources of biofuels. So government of India has initiated, and its future is to blend the present fuel with 15 to 10% with biodiesel, with bioethanol. And its source could be from agricultural bioethanol or from microalgae bioethanol.
There is one more question from Anjali Sharma. Sir, due to the presence of heavy metal ions and relative less amount of nitrogen and phosphorus in industrial wastewater, is microalgae a good solution for a large scale need?
So for less concentration of nutrients like nitrogen and phosphorus in an industrial wastewater, generally we go for the anaerobic digestion. So due to anaerobic digestion, the concentration of these nutrients like nitrogen and phosphorus got enhanced, because in anaerobic digestion we use different kind of biomasses like including industrial wastewater and then agricultural wastewater, and then it will ultimately increase your nitrate, phosphate, and nitrogen concentration. So we have to go. So this lecture was just we were giving a simple outlook of how we can use, but in practical purposes you have to go for different blending. So if you want to use microalgae for treatment of wastewater or wastewater as a culture media, then you have to first optimize how many nutrients are there. If some industrial wastewater has different less amount of nitrogen and phosphorus, then we will uh, combine this wastewater with some other different wastewater which are very huge concentration of nitrogen or phosphorus, like agricultural wastewater or anaerobic digest wastewater. So in this way, we have to approach for wastewater. Not only a single industrial wastewater or agricultural wastewater can be used, because if you go to in deeper depth, so you can see that wastewater also contains different kind of contaminants which are very harmful for microalgae. So first we have to go for optimization, whether this microalgae is suitable or it is providing any suitable growth in this wastewater or not. So we have to first optimize. We have to go for laboratory adaptation of microalgae. First we will adapt this microalgae at a particular strength of this wastewater. Then we will go for industrial application. So it needs different kind of optimization before practical or industrial applications.
There is a question from Manjari. Sir, if we use more than 10 to 15% bioethanol, then will there be any issue?
Sorry?
Like use of 10 to 15% of ethanol, will there be any issue? Like blending?
Because see, why we use 10 to 15%? Because these biofuels are very polar. And if you will increase the concentration of these biofuels, it will harm your engine. If you are, why we are using blend? Because in the current engine settings, we can, if you use 100% bioethanol, then it will corrode your engines. So if you want to use a engine which is totally fit for the biofuel, then you have to redesign your engine. So that's why government of India or any government in the world used to go for blending. So blending does two things. First, it removes burden from the fossil fuel, and second, it is if you go for its technical part, then it is very polar, and it will destroy your engine settings, corrode your engine if you will use in high concentration. Any other question?
Any other questions? If no more questions, then I would like to thank Dr. Nivas for joining us today and giving us an overview of microalgal biofuels and wastewater treatment using microalgae. Thank you so much, Dr. Nivas, for joining us today.
Thank you.